it's my great pleasure to introduce the next two speakers from the next talk. We are moving from something that was very interesting from the mathematical point of view to something that is very applied. So it's my great pleasure to introduce Dr. Heather Carleton, which currently serves as the Chief of Enteric Diseases Laboratory, EDLB, in the Division of Food and Waterborne and Environmental Diseases at U.S. Center for Disease Control and Prevention, also known as CDC. Her expertise lies on the field of enteric diseases, molecular epidemiology, bioinformatics, and next-generation sequencing. As the chair of PulseNet USA and PulseNet International Networks, she and her branch play a crucial role in connecting foodborne, waterborne, and One Health-related illness cases using genomics. The co-speaker is also Dr. Joe Wirth, which is a ORISE fellow that is working in National Antimicrobiology Resistance Surveillance Team, NARMS, and the Bioinformatics Research Development Team in the Enteric Disease Laboratory branch at the CDC in Atlanta. His PhD is in microbiology from 2019, and he did a postdoctoral scholars in the interdisciplinary competition program at the Harvey Mudd College in Claremont, California. His research interests include the application of computational techniques to microbiological problems, especially those involving the evolution of and physiology of human pathogens. Thank you very much for your time and the presentation. The floor is yours.
Well, thanks for inviting us to talk here about our work. I've worked with Joao for several years and learned a lot from him. Like he said, I'm more applied, so I use the algorithms that folks develop. I do not develop my own, but I'm going to talk to you about how we use phylogenomics for identifying potential foodborne, waterborne, or One Health-related outbreaks. And then my colleague, Joe Wirth, will dive more into some of our applied research projects to better understand better understand our strains, I guess. So I'll go ahead and get started. If folks aren't familiar with PulseNet, we detect related disease case clusters that may be part of widespread outbreaks. And if you've been watching the news in the US, one hand, a lot of it is about an election that may be happening today. And we also have been reporting a lot on an outbreak related to raw slivered onions that were part of quarter pounders served at McDonald's. And the sequence data that we generate and the analyses that we do helped identify that outbreak. So we provide real-time molecular surveillance of the most important bacterial pathogens in the US. So E. coli, Salmonella, Shigella, Listeria, Campylobacter, Vibrio, and Cronobacter. All of our sequence data is shared with limited metadata in real time to NCBI. And we coordinate within the United States to communicate about potential outbreaks we're detecting, as well with our partners around the globe. And I see some of our PulseNet International members are on this meeting as well, to communicate about potential foodborne outbreaks and help connect cases to potential sources. And if you're not familiar with the workflow of how we get whole genome sequencing into our PulseNet database, I'm just going to walk through it really quickly. When a person becomes ill in the United States and seeks medical care, a sample is taken and an isolate is generated. So one of our foodborne pathogens, that isolate is then sent to our state public health labs. So we have over 50 labs across the United States that then go on to perform whole genome sequencing and share their results with CDC electronically. We maintain a national database where we do searches using phylogenetics, so using single linkage trees to find in closely related isolates and assign them something we call an allele code. And we also do searches to find closely related isolates based on number of allele differences. Our primary analysis within PulseNet is core genome and whole genome multi-locus sequence typing. And then bacteria with the same fingerprint, our assumption here, similar sequence data are more likely to be linked to the same source. All right, next I'm going to hand it over to Joe to present the next few slides.
Yeah, so as I'm sure you all know, a lot of, really most of these types of treeing algorithms sort of start with a sequence alignment. Next. So one of those ways is a distance matrix where we collapse the alignment into some sort of distance matrix, and then we can build a tree based on that. Next slide. And one more. And alternatively, we could use the alignment directly, and these have pros and cons sort of for what we want to use them for. Next slide. So really the main treeing methods that we employ are either UPGMA or neighbor joining if we're using a distance-based approach. And if we're using a character-based approach, then it's generally maximum likelihood or Bayesian inference. And so we know, like with the distance matrix-based approaches, they can be reasonably accurate and they're significantly faster than the character-based methods. And if the matrix is right, then they can find an optimal solution. The character-based methods are generally more accurate under a wider range of conditions since they can include things like substitution models, but they're more difficult to implement and they're significantly slower. They don't scale very well. So the time complexity is too large to really find the best, to guarantee that the algorithm has found the best solution. Next slide. So really with these character-based algorithms, they are going to explore tree space, pick a tree, and then find the tree with the best score. But since tree space is very vast, next slide, it's a big O of, it's a factorial scaling problem. It means that, with only 100 tips, there's like 10 to the 100th more trees than there are atoms in the universe. And these scoring functions are basically saying how well does the alignment support the tree. And so because of this, if we want to look at the thousands and thousands of isolates that we are often interacting with, we sometimes will have to use the distance-based approaches like neighbor joining UPGMA. Next slide. So just, I guess, this audience probably is very familiar with this, but the maximum likelihood algorithm sort of is going to explore a subset of the tree space using nearest neighbor interchange. And because it can't explore all of tree space, it provides statistical support for the topology it generates using bootstrap replicates. Next. And then another model that we often use is Bayesian inference, and this explores tree space with Markov chain Monte Carlos, which is a way of sort of approximating the probability distribution. This is a little bit more in depth than I'm familiar with, but I'm definitely more of a user than a developer of this. But we can, instead of using bootstraps, we use posterior probabilities to provide statistical support for the topology that gets generated. Next. So here's sort of a practical example where we've used, this is a UPGMA tree. So the matrix that we used were allele distances, as Heather had alluded to. So this is sort of an abstraction away from alignment. It doesn't matter if there's one character difference or 100 character differences in the same locus, that would be one allele difference. So it's not perfect, but when these isolates are very highly related, it's a good approximation, and we don't have to do an alignment. We can just use a distance matrix based on allele differences. So we had identified a mutation in ESPW, which the ESPW allele is shown in the orange and teal track. So orange is a single base pair mutation, and the teal is the full length gene. And we sort of use that along with other epidemiological data to show that it was very strongly linked to a strain of interest of ours, which is the purple strain. So we were very interested in that strain, and we actually ended up revising the strain definition to exclude the ones that are gray in that same track. And it correlated well with the phylogeny we were seeing from this UPGMA tree. Next slide. And then we also used Bayesian inference treeing and time calibration on a subset of these samples. So we sampled randomly about 200 isolates and ran, I want to say, something like 500 million iterations. And we were able to sort of pinpoint that this strain of interest arose around December of 2015, which clinical cases were first observed in April of 2016. So that made sense with some of the data we were seeing. Back to you, Heather.
Great, thanks, Joe. And that's just an example of one of the strains we look at specifically for E. coli. In addition to outbreaks, we detect something that we call rep strains, which are occurring, emerging, and persisting strains. And then we work with our bioinformaticians like Joe to better understand the strain, if there's any unique markers that may provide hints to why we see it reoccur, emerge, or persist. In this example, it's a persisting strain, or maybe a reoccurring, one of the two. And the ESPW gene may be involved in making this strain have better survivability in the environment that we find it in. But in addition to this example with the rep strain, within PulseNet overall, we receive about 65,000 genomes a year. So about 30 terabytes of data that we have to analyze. And on here, our examples are for Salmonella, it's in the blue line because we receive about 55,000 genomes a year. For E. coli, it's closer to 4,000 to 6,000. And then for the other pathogens listed, you can see that it's kind of a scale, order of magnitude less than what we see for Salmonella. So we have to use multiple different approaches to identify potential outbreaks and to characterize our strains. Generating a tree with 55,000 genomes is not doable in a time frame for us to be able to detect potential outbreaks. And then over the course of a year within our PulseNet system, using whole genome sequencing, we detect around 200 outbreaks, majority of them Salmonella, but second most frequent is E. coli. And last year we detected about 37 outbreaks due to E. coli and 100 outbreaks due to Salmonella. So just to give you an idea of how many genomes we analyze and then how many outbreaks we detect, so potential outbreak clades. And about 10% of our genomes or isolates we receive are closely related to other isolates and associated with an outbreak. That other 90% is not associated with an outbreak and it is considered sporadic. So for whole genome sequencing, just to give you an idea of how we've been using it and how it's really revolutionized how we do our outbreak detection and surveillance, it really increases the amount of precision we use to be able to detect outbreaks. If some of you are familiar with the PulseNet system, we previously used a technology, pulse field gel electrophoresis, which was not as precise. And even though it detected outbreaks, the precision we get with whole genome sequencing is obviously on a different scale. It connects the dots for between cases that may be associated with an outbreak. At the CDC, we mostly detect multi-state outbreaks, and that may be linked to food sources that have a wide distribution chain. So having whole genome sequencing and being able to apply these phylogenetic approaches really helps us connect the dots on a foodborne outbreak. And then like what Joe is talking about, it helps us understand genetic evolution over time and how our different strains we track change and evolve. And then it really helps us target response by getting that confidence if we have a close genetic match between clinical cases and a food product, that most likely that food product could have been the source of the outbreak. So it helps us, it saves us time and money for targeting those responses. And so that's a quick overview of what we do within PulseNet and how we use phylogenomics. So thank you.